Hello, it is my pleasure to introduce you to the Translational Immunology program at St. John's Cancer Institute. This program initiated in June 2021 thanks to Rosalie and Harold Brown. And today I will walk you through what happened in these few months to enable the study of immunology in cancer patients. So first of all, I would like to explain why it is important to study immunology. So our immune system is really powerful. So in general, in normal condition, we have an arsenal of immune cells called innate immune cell, adaptive immune cell, humoral immune cells, whose job is really to defend us, to make sure that nothing bad or strange is happening in our body. It's like an army, more or less, and each of the soldiers has a particular role. And each of the soldiers to have that particular role require a particular characteristic. So our job is really trying to understand whether in the cancer patient we do have that army really complete, or if some soldier basically is missing, and if so, why is that missing? And our second job is to try to understand if we do have the soldier, so if we do have the right immune cell population, why in some case that immune population is not able to do properly the job. So what is actually missing, what is causing that? Is it something that comes from the tumor microenvironment? And for tumor microenvironment I mean like a sort of environment in which you have the cancer cell together with endothelial cells, normal endothelial cells together with immune cell, or is something that it comes from the immune cell per se. So the way the immune system works is that all these soldiers, they need to be aligned to conduct the job in a certain way. And if that doesn't happen, the things do not go right. And that's why some cancer cells are escaping and you have the spreading of tumor. Until basically the last 5-6 years, we were focused on studying the general characteristic of that of the immune system. So we were looking, for example, okay, how is the concentration of the innate immune cell in a particular cancer patient? How is basically the phenotype of a T cell in a particular cancer patient? But we were not really focusing on the details. For example, when we talk about T cell, when we talk about NK cell, when we talk about B cell, we don't have only one subpopulation. We have multiple subpopulations, right? So our job in the last 5-6 years, we are able to understand and appreciate that we need to go in those details. And in order to go in those details, we need cutting-edge technology. So we cannot just rely only on technology that are able to evaluate with one assay whether you do have T cell or NK cell or myeloid cell in the tumor microenvironment. We need basically more in-depth phenotype which are able to determine, okay, which kind of T cell, NK cells, myeloid cells, B cells you have in the tumor microenvironment as well as in the peripheral blood. So here comes basically the importance of translational medicine. So we need to understand those in-depth characteristics in real time from patients that are coming in the clinic. So we are collaborating with a number of clinicians at St. John's Cancer Institute. We are collaborating with the bladder program as well as the breast program as well as the melanoma program to make sure that we have in real time the appropriate samples which are needed basically to enable this research. So it's really a teamwork, so it's something that has to be done in matrix. There are different people who are accountable and responsible for the right sample collection at the right time, the right sample processing. So we are processing basically blood for isolation of a lymphocyte as well as plasma as well as serum, and but also for the processing of the tumor per se, like digestion of the tumor in a way that we can have like the separation on one side of the primary tumor cell as well as the immune cell. And what we do is that using different techniques and performing in ex vivo experiment, we are trying to understand what is the phenotype of those cancer cells as well as the phenotype of the immune cell. So it's a really a team job, and we are really grateful that with this gift we had the possibility to build all this, to basically implement a new team that is working together, but also to implement the like new technology. For example, one of the technology that we were able to here import at St. John's Cancer Institute is the DSP nanostring. And the second technology that we were able to bring here at St. John's Cancer Institute, thanks to this gift, is the spectral cytometry. So one, I can say that, so while the DSP nanostring is really able to characterize the tumor microenvironment from FFPE slides, so from paraffin-embedded slides, the spectral cytometry instead provides an overview of the immune phenotype as well as the cancer cell phenotype from the peripheral blood or from the tumor, the infiltrating lymphocytes. And why do I think that those technologies are really cutting edge? Because at the same time, both, they are able to focus not only on one marker, they are able basically to focus on different markers at the same time. So going back to what I said at the beginning, like we are really trying to understand not only like how is the harmony of the immune system inside the cancer patient, but in detail which kind of a characteristic those soldiers have. So do they express a particular marker which actually enable their activity? Do they express a particular marker that block their activity? So our spectral cytometry panel actually looks at together 25 markers. So really we are able to look at the whole immune system. Regarding instead the DSP nanostring, that also allows us to focus on the spatial distribution of those cells. So not only we are able to see what is contained in the tumor microenvironment, but how the linkage between one subset of cell with another subset of cell might influence the outcome of the patient. Thanks to the tumor immunology, we have an explosion, a revolution in the cancer treatment. So until 5 years ago, 10 years ago, we could have that the treatment for cancer patients were radiotherapy, surgery, and chemotherapy. Now we have a fourth pillar, which is immunotherapy. It's really working. It's really set up the stage and improve the cancer patient life, particularly in some patients, such as patients with melanoma. But are we able to say that we are 100% over the cancer fight? No, because still some patients do not respond to such treatment. So there is still the need from our side to understand what else can we do and how we can do that. First of all, like we need to learn how to interpret those big data coming from this cutting-edge machine. We need to train the system, so we need to really to improve the data analysis and foster artificial intelligence in a way that we are able to look at the problem in the right angle. So I do see that our mission is not complete at all. I do see that we do have a lot of work still to do because there is still so much to understand in terms of like why some patients are responding, why some patients are not responding, what is the appropriate combination therapy, how do we interpret the data, how do we foster basically artificial intelligence. So this is our mission. This is basically what we need to aim to do in the next years. And to do so, we cannot pretend to do that by ourselves. We really need to build collaboration, a great team, trust among the team. So you saw, for example, different people in this video. Some researchers are more focused on some cutting-edge technology, others instead are, for example, Alberto Mendoza is a postdoctoral fellow who is very skilled with flow spectral cytometry and flow cytometry as well as molecular immunology, came straight from Spain to join this lab. We are really lucky to have him. We do have also Kyugen, who is really skilled in isolating tumor-infiltrating lymphocytes. She worked in this field for more than 15 years, so we are really lucky to have her. Gary, who is processing this blood sample for us, and also worked at St. John's Cancer Institute since a long time. He helped basically Donald Morton also to build the biobank, the huge biobank of samples that we have at St. John's. So we really need to make sure that this team is empowered to do in the best way their job. And my mission is to make sure of that, and to make sure that we don't stop, we move forward in order basically to allow cancer patients to live more, to fight cancer.